membrane here, lateral plasma membrane. So you can almost see 1, 2, 3, 4, 5, 6, 7 cells here, conical cells. It's a coalition of 7 cells. And all these sexual granules are being released into the lumen. Okay, you can see them at this apical portion. Well, maybe this picture gives you more idea about it. So these are type 1 granules. Now, by the time these type 1 granules are fully manufactured and when they are being released into the lumen, immediately the type 2 granules begin to form. But what we noticed is this type 2 granules has different morphological structures. So we just want to name it as type 2A, B, and C because we see these 3 very distinct, significant changes in this second type of sexual granule. So this sexual granule, as you can see here, is very dense. This is very, very prominent compared to the type 1 granule. So this is another type of granule that's forming inside this cell. And as the time proceeds, this is how the cell looks in the breeding season. Now it's completely hypertrophic. You can really, really appreciate the length of the cell, completely conical shape, the broader base and apical lumen. The lumen is very narrow, right? And the secretion is so much filled in the cytoplasm that even the nucleus is pushed to the basal portion. And that's very, very clear here. And what is another very interesting factor here, there's numerous mitochondria being formed. All these structures are mitochondria. So as and when the demand of the cell is increased, the right heroes comes into picture, isn't it? Mitochondria is very, very essential for the maintaining the bioenergetics of the cell, and that's exactly why you see the abundance of mitochondria here. And you can see the sexual granules at varying apical densities, sizes, and locations. Now, what is even interesting, we also noticed that, well, I did not bring this in the materials and methods, but just want to let you know that we parallelly studied the circulating testosterone concentration. So what we know that the circulating testosterone concentration, that is the serum testosterone concentration, goes in parallel with these changes. So it is twofold increased in breeding season, I'm sorry, in the regenerative season. It becomes threefold increase in the breeding season and slowly it drops down. And what is very interesting that we couldn't actually make sense and no literature makes mention about it. Something which is very, very paradox here is that these electron-lucent crescent-shaped bodies that we noticed inside the nucleus. Now, what you're seeing here is a complete portion of a nucleus, and this is the nucleolus. You can very well appreciate the nuclear membrane here. But this, electron-lucent crescent-shaped, half-moon-shaped structures, we don't know what these structures are. As I told you, there is no literature support for it. But we know that this particular cell is tremendously under the influence of testosterone now.
Did we lose the audio?
Seems like, sir. Dr. Sampson, I think you are on mute and you are continuing to talk. Please unmute. We may have lost connection. Sir may join back. We will just wait for a minute.
I'm trying to connect. I'll find out.
Okay, am I audible now?
Yes, sir. Yes. Please continue.
Okay. Is my slide visible also, madam?
It's visible, sir.
Sir?
Please continue.
Okay, thank you. Thank you.
Just a second, sir. Yes, ma'am.
He's visible and he's audible also.
Okay, thank you. Okay, that's quite relaxing now. Okay, this is the type 1 secretory granule. As you can see here, there's the Golgi vesicles are trying to pack it. Now, why am I stressing on this point here is we later found out, we did some biochemical estimations and found out that this is basically a carbohydrate motif. So it has a lot of carbohydrate contents here, and therefore we see this form of packing of this Golgi here, and the same thing is happening with this type 2 granule. Now, I'll quickly explain what this type 2 granule is and how it changes. Of course, you can very well appreciate this nuclear changes happening here. Now, this type 2 granule, which had this humble beginning, now shows a very dramatic changes on its surface. Now, you can very well appreciate that here. So the one which started like this shows now all these electron opaque patches here on its surface. And another interesting character here is they don't merge with the neighboring granules. They maintain their integrity. And all these granules, by the way, they are released in the process called apocrine secretion. Now, what is apocrine secretion? Apocrine secretion, where there is a loss of apical plasma membrane when these granules are released. As you can see here, this apical plasma membrane is slightly torn, and the particles or the pieces of apical plasma membrane you can see here in the lumen, and that's how this heavy cargo is put into the lumen. The third type of granule which we noticed is completely dense. It is completely different from type 1 and type 2, and so we named it as type 3 granules. These are very, very distinct granules, and they do have this electron lucent core and electron dense rim around it. Okay, apart from that, what we could see here is these type 3 granules, as they age, they do have the shriveling appearance, and slowly they degenerate and they release the content into the cytoplasm. Now, this is all the events that's happening in the breeding phase, and as the non-breeding phase occurs, the whole drama changes. Now, the cell reverts back to its earlier condition. You can see a less number of mitochondria, and you don't even find a single sexual granule here. The cell is completely filled with mucoid substances. Now, how do I conclude this? Well, the granules that we just studied, they have a greater relevance in reproduction. Now, what are some of the functions? Now, as you can see here, I have just listed out a few functions here. Now, these granules are very, very important in sperm sustenance and activation. They provide the biochemical energy in terms of dissipation of ATP all along the length of the tail of the sperm. And it also includes or contributes to the nourishment of the seminal fluid. So it provokes seminal fluid production. And very interestingly, these granules form a plug in the vagina of the mated female, so that whatever sperm has been deposited should not leak out of the vagina. And this is a very rare phenomenon in case of lizards, but it's quite common in case of snakes. And also it supports capacitation. And very interestingly, these granules can even cause uterine muscle contraction to facilitate the movement of sperm inside the female body. And it also is a flag. It produces pheromones so that it can attract the mate during the breeding season, because here the mating is a very, very costly affair. There are serious battle, fierce battle for sex. So therefore, all these gifts are very, very essential to have a successful mating. And another important function of these granules, they form spermatophore. Now, the spermatophore are the very thick, cloudy ridge that's constructed against the around the walls of the vagina in case of female, and that has greater relevance in sperm sustenance, motility, and at the same time it supports capacitation too. Now, why is that we have to study these granules? Well, this is very central to understanding the biology of reproduction in case of squamate reptiles, because I would like to bring this last point here in this presentation. Studies have shown that if you chemically try to abolish these granules, okay, even though the testis is fully functional, even though the testis is fully loaded with sperms, it is as good as the male lizard is completely infertile. It's as good as a vasectomized male. Okay, so these sexual granules are very important. Now, one point that I have to make it clear is now these sexual granules are happening in the kidney, the nephrons, And how is that exactly, how is this conveyed to the female genital tract is the question. So what happens here is that there are a lot of ampullary connections, there are ductal connections, and these secretions will pass through a common opening, a common sphincter, and then finally it reaches the cord of the hemipenis, that is the penis of the lizard, and finally it mixes with the seminal secretions from the seminal glands, and thus it is expelled into the vagina of the female during mating. So understanding this gives us inputs about the complex nature of biology of reproduction in reptiles. I'll be happy to take a few questions. Thank you.
Thank you, Dr. Sampson, for that very lucid and clear presentation about the renal sexual segment of Eutrophis carinata. I think the work is very interesting, and I think it threw light to most of us how these granules play a very important role in the fertility of the male lizard. I have just one question, Dr. Sampson. What is the IUCN status of this particular Eutrophis carinata?
Well, it's actually least concerned. So in the year 2011, we had a green signal to work on these lizards from CPCSEA and the forest, the desk of the forest conservation. But the status of this particular species in other parts of Asia is now a bit of concern, madam.
Okay. So how did you procure these lizards, doctor, for your study?
These are wild-caught lizards. My study area was, again, the woody areas surrounding the University of Mysore. And we had a very restrictive geography. We couldn't go beyond that because of the rules and regulations of CPCSEA. So we used to wait for these lizards. They are very shy, by the way, and they are subterranean in nature. So we used to wait for these lizards, and we used to collect just one or two lizards, and that's how we did our study. And a larger platform of correlation was done based on the subsequent literature studies.
Okay, okay. And what are the granules actually made up of? I'm sure you've done some biochemical work.
Excellent. Yes, ma'am. Yes. Interestingly, these granules, and all these three granules are enzymes, and that was for our shock. Enzymes being produced in this colossal quantity is definitely a shock, and that's exactly why I always say that the herpetology or this reptile biology is a land of treasures. It sometimes takes you away from the normal or the cliche of understanding that we have. So all these enzymes, one of the predominant enzyme is GAPD2, glycerol 2-phosphate dehydrogenase. We could also locate this enzyme on the matured sperm, and we could also see that in the absence of this enzyme, the tail of the sperm doesn't show a whip-like action. The sperm can still move, but it cannot move much faster. But with the presence of this enzyme, the sperm can move faster and it can reach the target early.
Okay. Thank you, Dr. Sampson. There's one question in the chat box. Sir, what's post-breeding?
Post-breeding is a very short phase where you can still see the remnant granules in the cytoplasm. Okay, the granules are still there, but these granules are somehow inactivated. Okay, one thing that we have to appreciate is this whole event is strictly very, very punctual, and it really fits into the time frame. So these granules, the granules that we see in post-breeding are the granules which are manufactured much later in the process, so they don't get a chance to mix with the sperms. So they remain in the cytoplasm, and finally they disintegrate. So post-breeding simply means where you can see the granules in the cytoplasm, but they do not have any biological relevance. Their fate is to just die within the cytoplasm itself by the action of various lysosomal enzymes.
Thank you, Doctor.
Thank you, Dr. Sampson, for that wonderful drift from ecology to histology and physiology. Thank you so much. We have now Fanai talking about the survey on the anuran diversity in and around the Reek Community Reserved Forests, Mamit District, Mizoram, India.
Ma'am, I will be presenting from here since my
Yes, sir,
laptop is
sure.
not working.
Please. No problem, sir. Please continue.
Is it visible on my screen?
Yes, sir. It is visible.
The Community Reserve Forest is located within Mamit District, Mizoram. The edible ranges of the surveyed area is ranging between 515 to 1,485 meters. So this area has been a locally protected land since the 1890s. So our present work, we try to perform the documentation of the diversity as well as the habitat of these anurans within the reserve forest. And so the temperature ranges in summer. It also receives a good amount of rainfall during a period of 10 years, which is 2681.97 mm. The soil is composed of slate loam in the peak region, while the rest of the area is mostly sandy loam with black humus topsoils. The study set does not enclose any major streams, but there are about 7 small seasonal brooks and streams, which all flows into the Tlong River. The vegetation can be classified as semi-evergreen forest or subtropical evergreen forest. The forest has been managed by the local people uh, through selective felling. Like for example, when they uh, subject some lands for harvest, they would leave some mature trees for the population of the plant fauna. Flora, sorry. So these are the habitats where we do the survey. As you can see, and here, these man-made water bodies, they are excellent breeding grounds for the anurans. So we recorded 21 species. Um, so the species collection was done through visual encounter survey and acoustic encounter survey. The survey period is from January 2019 and is done till date. Day and night surveys were conducted within the reserve forest with its surrounding area, covering grasses, shrubs, crevices of rocks, leaf litters, plantations, forests, streams, and water reservoirs, including like temporary ditches, pools, and potholes. Adult frogs are captured by hand after locating their habitats, and tadpoles were also collected with the help of a scoop net. So the collected specimens were fixed in 10% formalin and are preserved in 70% ethanol in the Amphibian Section, Departmental Museum of Zoology, Mizoram University. So going to the results, during our survey period, we recorded the presence of 21 anuran species belonging to 6 families. From the Bufonidae, we have the Dotaphrenias melanostictas, and the Dicroglossidae is represented by the Euphyctis class Graminensis and the Minerva Asmati. From the Megafish, we have the Megafish, and from the Microhylidae, we have the Colloidal Bullfrog, Microhyla Birdmeirai, Microhyla mukhesuri, and the Microhyla aishani. And the Ranidae is represented by the Amolopsindo permanensis and the Silvirana lacrima, and the Archophoridae is represented by 10 species, including the Kuriselas Yangi, Orchestis, Rakzakani, and Manipurensis, the Polypedatis braveri and terraensis, and the rare Telederma bibungens, molog, and the nagalandans. We also encountered the Zanges Maraguinas and Zanges safri. So I will give a brief account on the species we encountered. The first one is the Dotaphrenias melanostictas. It is known as a common Indian toad, and they are characterized by their distinct tympanum and the presence of a yellow dorsum, which can also be red, brown, or gray, and is used to identify the species. Next, we have the Ephilictis calasgramensis. These are commonly known as the Indian skipper frog or the skipping frog, and they are mostly found in ponds, and their species distribution covers most of the Southeast Asia. We have the Minerva Asmati, known as the pedirized frog. They are a small size or medium-sized frogs of the Indian wetlands. The species is widely distributed in India, Pakistan, and Sri Lanka, through the Southeast Asia to China and Japan. We have the Megafish Awoo. This is a medium-sized frog with head broader than long, prominent nostril close to the tip of the snout. Dorsum heterogeneously dark, tip of snout concave dorsally, upper eyelid with two prominent warts. Ventrally, the throat and the chest are brown to black. Their microhabitat includes the leaf litters and are encountered mostly on the way to the peak of the mountain. Their distribution is confined within the Northeast India. And we have the Megafish cercipi. It is also a medium-sized toad-like frog. The head is broader than long, nostril prominent, close to the snout tip than it is to the eye. They are found throughout the study sites. Next, we have the Colloidal Bullfrog, known as the banded bullfrog. They have a rounded body with a generally brown coloration with whitish stripes. They were found in temporary pools along the streams of the forest with an elevation of 1220. Their distribution included Northeast India, China, Tibet, Bangladesh, and Myanmar. Next, we have the Microhyla Birdmeirai, commonly called as a narrow-mouthed frog. The species is generally yellowish in color with dark brown patches. The characteristic feature of this species is the absence of teeth in the upper jaw, and the skin is smooth with a small head, an oval tongue, and a vertical or circular pupil. Next, we have the Microhyla muclasuri, known as the muclasur's narrow-mouthed frog. Its surface is marble-like and is V-shaped with a distinct V-shaped head. They are found within China, Myanmar, Bangladesh, India, Thailand, Indochina, and Singapore. Next, we have the Microhyla aishani, named after the northeast region of India. It is strikingly different from other narrow-mouthed paddy frogs by characters such as reddish-brown coloration on the back, prominent dark streaks and ash-gray moting on the lateral sides, and the absence of webs on its feet. These frogs were encountered near human settlements. Their distribution included northeast India and adjacent Myanmar. We also have the Amolops indobermanensis. It is commonly known as the Burmese cascade frog. They are tan brown, olive green, or olive brown with well-defined inky black spots on the back. They are mostly found on streams with fast-flowing waters, and their distribution included Myanmar and India. We also have the Silvirana lacrima, commonly known as a cricket frog. It is a medium to a large-sized frog. They are widespread in the Southeast Asia and occurs widely in the peninsular Malaysia, Borneo, Indonesia, Philippines, and India. Next, we have the Kuriselas Yangi. It is a small tree frog, and the female is larger than the male. The head is broad with a triangular pointed snout. They are found mostly on the grasslands and among leaf litters inside the secondary forest. We have the Orchestis manipurensis and the Orchestis Rakzakani. They were collected both from bushes and from leaf surfaces, which are about 1 to 2 meters above ground. This Orchestis Rakzakani is only known from the type locality in Bangladesh and has been formerly reported from the Dampa Tiger Reserve, Mizoram, India. When we have the Polypedatis broweri, it is known as a white-lipped tree frog, and its head is more longer than its width. The cephalic skin is loose from the nasal and frontoparietal. Snout is pointed and projects beyond the lower lip, little less than half of the head length. They are found on grasslands, muddy paths, roadsides of secondary forested area with an elevation of around 1200. And here the Polypedatis terrensis. It's also known as the bamboo tree frog. It is a medium to a large sized tree frog which is brown to tan on the back and the side of the head is darker and legs are cross-barred. They are distributed throughout the Southeast Asia including India, Nepal and Bangladesh. Now we have the Teloderma bibugens. It is a small frog with a snout vent length of about 25 to 35 millimeters. The head is wider than the body and slightly wider than long. The snout is rounded and slightly longer than the diameter of the eye. The nostrils are closer to the tip of the snout than it is to the eye. We have the Teloderma molog. It is also known as the Indonesian tree frog. It is a small-sized frog with reddish dorsals spotted with white on the ventrals. They are arboreal and are found on trees and has a very good camouflage. They are found mostly on moist environment and are isolated in their microhabits. We have the Teloderma nagalandans. It is also commonly known as a Nagaland tree frog. It is a small sized frog and the body is covered with tubercles and the tip of the fingers and toes are red in color. They are arboreal frog and are found on cinnamon tree near Tourist Resort with an elevation of around 1,196 meters. Next, we have the Zanges Maraguinas. It is a giant tree frog, green in color, with protruding eye and toes are properly webbed. This species is widely distributed in Asia, including India, Nepal, and Bangladesh, and throughout Southeast Asia. And lastly, we have the Zangizelas safri. It is also known as a flying frog, and it is a small tree frog-like appearance, having a red-colored web on the hind feet. The species was found on the road on a rainy day in the month of August. Their distribution included Mizoram, Assam, and Bhutan. And from this study, we can conclude that the microhabitats of different species vary and most were found on roadsides, vegetations inside the reserve forests, muddy paths, leaf litters, drainage canals, and water storage tanks. The spatial distribution of the species greatly varies depending on their morphology and adaptive features. For example, Minerva Asmati was found almost on all regions of the study sites, whereas Amelops indopermanensis was found only in streams region, which consists of fast-running waters, waterfall, bedrocks, and boulders. From this study, it is clear that anurans thrive on different ranges of elevation. Some are found only on highly elevated areas such as the Orchesis and the Megafish, while some species were encountered only in one elevational range. Some also thrive in all ranges such as these Zangizelas maragdinas. The number of anuran species reported in this study represents about 4.43% of the total species of anurans recorded from India. Two species, the Teloderma nagalandans and the Megafish Nambumayang, this should be Megafish Awoo. Sorry for my typing mistake. These were reported from Mizoram for the first time, and the occurrence of Kuriselas Yangi from India was also recorded from this study. And I would like to thank the Principal Chief Conservator of Forests for the research permit and for the National Mission for Himalayan Studies for providing the funding for the work. This was for my presentation. Thank you.
Thank you very much, Kanai. Preeti ma'am.
So, Vaila. Okay, thank you.
Yes, ma'am.
Kumar, for your presentation on the various species of frogs which you have found in your region of study. I had just one question, that family Rhacophoridae had the most number of species, I think, from the data
Yes,
what you have. Yes. Anything connected, like why that particular family was more, or do you have similar literature or similar research work in some other area also to show that Rhacophoridae had the largest number of species recorded?
The number of the rhacophorids being higher than the rest of this family can be attributed to the diversity of the habitats found within the study area.
Okay. Okay, so they thrive in more types of microhabitat compared to the species belonging to other families. Am I right?
Yes.
Yes?
Yes,
Oh,
ma'am.
okay, okay. Thank you, Amit Kumar.
Thank you, Fanai and team, for the wonderful presentation. There seems to be no questions from. Oh, no, there are a few questions. Sir, I heard that people consume bullfrogs. Is it the same as you mentioned? There's a student asking this.
Mm, from our region, we have recorded that people consume this Silvirana lacryma or the alticola, but I don't think we have any report on the consumption of bullfrogs from our region.
Okay, thank you very much. That was the only question that was there from the audience. We appreciate all your hard work and your presence today. Thank you so much. Dear participants, there is a link on the chat box. Kindly fill in the details for your attendance. It is mandatory. Since you attended the seminar, please fill in the details there and submit it for your attendance. Thank you very much, Dr. Seethavi.
Yeah, okay. Thank you, Dr. Viola, and thank you, Dr. Jaishankar, and the organizers for giving me an opportunity to share this session.
Thank you, ma'am. Thank you both, Dr. Sattvi, Etinder, and also Dr. Viola Mendons for chairing the session. We had 4 oral presentations in this session. With that, we have come to the end of the first day of the 2-day national webinar on herpetology, Indian scenario. I now hand it over to Selva Priya, one of the student coordinator of the webinar, to render the vote of thanks. Thank you.
Acknowledging the good that you have is the foundation for all abundance. On behalf of all the members of the Natural Science Association, I extend my sincere gratitude to our college management and our beloved principal, Reverend Father Victor Lobo, for helping us put our thoughts into action. I extend my gratitude to the zoology department of our college and all our professors who have been encouraging for the success of this event. Thank you to our speaker, Dr. Dinesh, who readily shared his knowledge and seeded our interest in herpetology. A very big thank you to all the oral presenters who came forward to showcase their works. I wholeheartedly thank our chairpersons and the moderators of the event who were a pillar of support throughout. We are forever grateful to our Natural Science Association coordinator, Mr. Jaishankar. Thank you, sir, for all your efforts, ideas, and support. I also would like to thank all the co-members of the National Science Association and all the volunteers who worked together to make this event successful. Last but not the least, I thank all the attendees who spent their time with us in gaining knowledge. Thank you, everyone. Hope all of you all enjoyed your time with us.
Thank you, Selva Priya. Selva Priya is one of the student coordinator along with Rakshit, Basil, and Andrea. With all their efforts, that has come out well in completing the first day. Please join us tomorrow as well. The session starts at 4 and hopefully finish a bit earlier. Today it was supposed to get over, but because of the technical glitches. Also, we would like to place on record our chief guest, registrar, for the chief guest address. Coincidentally, in most of the events, the principal of the college is the chief guest. And today it happens that the registrar was the acting principal. So NSA had the principal in being the chief guest. Thank you all. Good night. See you tomorrow at 4. Please join for the second day of the national webinar on herpetology Indian scenario. Good night. Take care.